I'm Paul Soudier from University of Minnesota. I'm on a long-term HFSP postdoctoral fellowship, and I'm studying bacteriophage. So what is bacteriophage? Basically, so these are virus that infects bacteria, and they do actually represent the most prevalent biological organism you can find on Earth. So they are displayed this very wide diversity of structure, very wide diversity of genomes, and among this diversity, many of them use this, what we call lytic phage infection cycles, which can be summarized this way, which is very central. So you have adsorption, you have DNA injection, DNA replication, and then it produces new phage particle and eventually lyse the cell. So this has been very largely studied, especially during what we call this golden age of phage biology, which was from the 40s to the 70s, with this whole group of phage biologists that study that, and also on the side discover the basic rule for molecular biology. One good thing, actually, after that, we would know most of what's to know, especially for the model phage, but some knowledge gap do actually remain, especially concerning quantitative aspects. And digging into that, I've been actually surprised to find this very famous a quote from this famous Nobel Prize laureate, which is Arthur Kornberg, that stated that in 1974, it was very interesting for them to try to rebuild this whole viral life cycle in a chemically defined cell-free system. And I was surprised to see that it hasn't been done yet. And to me, I'm not really a phage biologist, but I'm a cell-free system biologist. And so it was very interesting to me to work on that specific system. So just briefly, cell-free system, what it is. So the ones we work with are like lysate-based cell-free systems. You take a bacteria, you make a lysate from that, you supplement various chemical molecules, and you get a system where if you provide DNA, you can actually make transcription and translation in this in vitro system and get proteins. What is super interesting is that in the last 50 years, there have been tremendous advances on this system. In particular, in the 2010s, some people demonstrated that with a good enough cell-free system, you can actually put phage genome inside this reaction, and this whole system is actually able to do transcription of phage mRNA, translation of the phage protein, and they can assemble and encapsulate the genome to make active phage particles. So this is one side. The other side of the advance have been all these people working on creating synthetic cells with cell-free system inside. So I won't detail the process, but basically you can encapsulate that in a custom lipidic membrane. So this was really the starting point of the project. And so in that project, what I was focused on was building some synthetic cell displaying phage receptors. So in this case, it was LPS synthetic cells, and building some specific phage able to infect these. And when you mix these together, what you see is you have this nice red fluorescent signal indicating that you do have this infection. So if you want to see more on that and have the full story, you can see me at the poster 25. Thank you.
Thank you.